Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Afif Farhan bin Hosnaini. Today, I will make the presentations about the structural skeletal muscles and the sliding filament theory. Before I start my presentations, basically my presentation is based on the slide that given by the Sir Shafiq. Okay, first we can start. Firstly, the skeletal muscles is consists of many things to form a skeletal muscle, which is it is consists of fascia, fascicles, epimysium, perimysium, endomysium, sarcolemma, myofibril, sarcoplasmic reticulum, and the sacrums. Okay, for the first slide, which is the picture of the structure of the skeletal muscle, we start from the fascia. Okay, what is the fascia? Fascia is a connective tissue that wrap all internal part of the body, included the muscles. From the diagram, we can see the muscles is surrounded by the layer that called epimysiums. The epimysium is the outside layer. Okay, then we go to the fascicles. Basically, the fascicle is a connective tissue and a bundle of muscles to form the muscles. To form a muscle, a bundle of muscle tissue should form first to produce muscles. The fascicle is surrounded by the perimysiums. The perimysium is located at the inner part from the epimysium. From the diagram, we can see a layer. The layer it is start from the epimysium first, which is the outside, then following by the perimysium, which is surrounded at the bundle of muscle fiber, which is the fascicles. The endomysium we can found at the each of muscle fiber, because the endomysium is surrounded at each muscle fiber. It is different from the perimysium because the perimysium surrounded all the bundle of muscle fiber. However, the endomysium is surrounded only each of the muscle fiber. In this slide, it is the same what I have mentioned in the previous slide. It is basically the same thing, which is the epimysium, the perimysium, the fascicle, and the endomysium. Okay, then we go to the next slide, which is the microstructure of the muscles. We can see in the microstructure of the muscles, we have the sarcolemma, myofibril, sarcoplasmic reticulum, and sacromeres. Okay, from this picture, we can see a bundle of skeletal muscle fiber is extend. So, from the skeletal muscle fiber is extend, we can see the skeletal muscle fiber is consists of the myofibrils. Okay, the myofibrils, in the myofibrils we can find the myosin and actin filaments. The actin and myosin filament is different to each other, because the actin filament is a thin filament, however the myosin filament is a thick filament. From the name, we can also differentiate which is the actin thin, which is the lipase. Okay, then we go to the sacromeres. The sacromeres is basically a structure unit of the myofibrils in the sacromeres. The myofibrils will further the subdivided into the individual segments. The segments means in the sacromeres there is a light and dark band and also has the Z line. Okay, for the sarcolemma, it is just a cell membrane surrounded the muscle fiber. Now we go to the important part in the contraction and relaxation of the muscle, which is the sacroplasmic reticulum. Okay, the sacroplasmic reticulum is very important in the contraction and relaxation of the muscle because because in the sacroplasmic reticulum it is store the calcium ions. Okay, without the calcium ions the muscle contraction cannot be occur. Okay, from the picture we can see there is the nucleus, which is the nucleus is the function to control all the activity mechanisms, and the mitochondria. We can see the mitochondria. The function of mitochondria is to generate the energy, for the contractions and relaxation of the muscles can be occur. Okay, in these slides, I also have been explains about what is the sarcolemma, myofibrils, sacroplasmic reticulum, and sacromeres. Basically, it is the same thing that I have explaining before that. Next, I will explain to you about the process of sliding filament theory. Okay, from the slide we can see the actin and myosin myofilaments in a relaxed muscles. The myofilaments A band do not change length during the muscle contraction. So in this picture you can see the structure during in a relaxed position. It means the person does not do any movements. There is no any muscle contraction occur at that time. So we can see the A band, which is you can see does not change their length, and the Z disc is at their normal pace, which is does not change because there is no movement occur. Okay, for the next slide is about the during contractions. Okay, during contractions, actin myofilament at each end of the sarcoma slide past the myosin myofilament toward each other. As a result, the Z discs are brought closer together and the sarcomere they shorten. Okay, it means during the muscle contractions, you can see from the picture, from the slide, the actin myofilament that is in a purple color. Imagine this is a actin myofilament, and the myosin myofilaments that is in the green color. During the contractions, this myosin and actin filament will slide past toward each other. Okay, because of they slide at each other, so it will make the Z disc. You can see the Z disc from this area and this area. During contraction, it also will move closer to closer at each other because of the muscle contraction being occur at that time. Okay, for this slide, it state that as the actin myofilaments slide over the myosin myofilaments, the H zones and the I band start to narrowing. Okay, it means during the contractions, the actin and the myofilaments start to slide over to each other. So the H zone, which is in the slide, you can see the H zone is in the brown color, start to narrow, and the I band also start to narrow because during contractions our muscles start to be shortened. So at the same time we can relate that the H zone and the I band also should be narrowing because of the contractions. Okay, then the air band, which are equal to the length of the myofilaments, do not narrow because the length of the myofilament does not change. Okay. Okay, in this slide, you can see the structure when the fully contraction is happens. Okay, from this slide, it say that in fully contractions muscles, the end of the actin myofilaments overlap and the H zone disappear. Okay, imagine before the fully contraction occur, the actin this is the actin myofilament, this is the actin myofilament. This is before during contractions. So during and there is a H zones at the middle. Imagine it. Okay, when during contraction will occur, the actin myofilament will overlap with each other and make the H zone at the middle is disappear when the fully contraction occur. It is because during the fully contraction, it means the muscle is totally short, so it will make the H zone disappear. Okay, this is the last slide. Okay, this slide is basically is summary of the process of sliding filament theory, which is where it start and how they start and how they finish. Okay, I hope my my explanation can make you all understand a little about the sliding filament theory. Okay, thank you.